Before I begin, since during my PhD in Boston, I want to first acknowledge the land and water Boston occupies. These are the unceded, ancestral, traditional, and contemporary lands of the Massachusetts, Pawtucket, Wampanoag, and Nipmuc tribal nations. I honor the cultures, contributions, and enduring presence of these native peoples. At the same time, I call for us to be better stewards of the land and allies to Indigenous communities, committing to learning from and supporting Indigenous leaders. It is essential to move beyond acknowledgement towards action, working to dismantle oppressive systems and elevate Indigenous voices and stories in the community and beyond. So with that, I will begin my presentation, which I will talk about my dissertation work in advancing the methods for studying the neuroscience of sex variability and diversity. So my PhD explores sex, but what is it actually? When the historian Beans Philocci asked the undergraduate class, this was something that they came up with, which included, but not limited to, the chromosomes, the genitals, appearance, sex partner, which indicates this multiplicity of this conceptualization of sex. And throughout history, this has changed over time, that have accrued an increasing number of conflicting yet coexisting meanings. Despite this, most studies actually examine sex as a singular and stable entity, but in actually, it is more like a proxy, an imperfect heuristic of the variables that are of interest. It is also unrealistic to think that individuals fulfill the archetypes of all variables relating to sex. Jacques Derrida articulated deconstruction as a way to challenge the texts and meanings that never cohered in the first place. And several frameworks has been proposed to deconstruct sex that challenges this dominant rhetoric towards more of sex being not binary nor essentialist. And this diagram demonstrates this using the rodent receptive behavior, which is known as lordosis, as an example, where you have this different scales of sex-related variables that varies, and there's also the dimension of time. And in this diagram, only some, but not all of the possible covariates and functional relationships are present, where the ones highlighted in black are just example studies that test the hypotheses of these one of many possible functional relationships. These are also dynamic and are affected by many contexts, such as development, experience, and the environment. This summarizes the multiple considerations when deconstructing sex, with the ones in the purple box are the ones relating to experimental methods and theoretical basis, such as the consideration of non-sex variables as causative explanations instead of just sex. It is also to recontextualize how inferences are made from studies on model organisms. The boxes in teal adds the social input and output, for example, to acknowledge how cis-heteropatriarchy in research have affected the evidence obtained, because science is an active construction of the world that is used for public discourse and standards that have affected sex-gender diverse individuals. So my dissertation is shaped around this framework of sex being multivariate and multidimensional. And with that framework, I worked on advancing methodologies that could help with constructing sex variability and diversity, where variability refers to the different sex-related variables within a species, and diversity refers to the sex variability across the species. And these are specifically pivoted to strive towards a more just and equitable outcomes for sex-gender diverse communities. So with that, these were manifested in these 4 aims, which I will take you through one by one in this presentation, where it spanned across the scale of sex-related variables and across the different species in this more than human world. So going now into dissertation aim A's background. To begin, I want to address this issue of our lack of tools to study the molecular, cellular, genetic aspects of human sex variability that is easily accessible and physiologically relevant to diverse individuals. As an example cell type that could help advance human sex variability are sebocytes, which are these skin cells that are primarily involved in the production of lipids that protects the skin. What's more is that they also communicate with the nervous system as they synthesize the several neurohormones, as well as expressing their receptors. They are also known to be very androgen sensitive, which relates to sex. To develop human sebocyte models, we could leverage induced pluripotent stem cells, iPSCs, which can be generated from non-invasive biological samples, such as from your hair, your skin, your urine, that can then be reprogrammed back into this embryonic-like state where they can be differentiated to any cell type. They also have the advantage of retaining the genetics of the donor, which allows individual-specific studies to be conducted when adding molecular and environmental factors relating to that individual. The expansion of sebocytes from iPSCs could also benefit from less worries of a limited lifespan and genomic instability. So in this section, I aim to engineer human sebocytes from iPSCs that can be used to study the cellular, molecular, genetic aspects of sex and their interactions with neuroscience. To differentiate iPSCs into sebocytes, my strategy was to directly activate transcription factors, TFs, that drive cell fate programming. And I could control the TF expression by delivering this type of DNA that can be integrated into the cell genome when this specialized enzyme is present. And these DNAs contain TFs that will be expressed in the presence of the drug doxycycline. And after several days of treating the cells with the drug, I could isolate the cells that are low in TRE160, which is a marker to determine whether the cell is still a stem cell or not. If it is low in TRE160, this means that the cells have differentiated. Then I will measure the gene expression of the differentiated cells at the single cell level to determine if they match human SIBO To perform these experiments, I curated a list of TFs that may relate to the SIBO lineage. I transferred all of the DNA where each one of them carry one TF and transferred these into the iPSC lineage, where each individual cell could take up any number of TFs. Currently, it is not possible to determine a priori which combination will result in SIBO. So this is part of an iterative engineering design process to refine a combination that works. And what this process means is that not all of the iPSCs will differentiate. So I determined which cells differentiated by measuring the Tre160 levels. And to do that, I first isolated my samples as cinglet cells and then selected the cells that are low in Tre160 for sequencing. After single-cell RNA sequencing of the differentiated cells, we analyzed the gene expression. And we used a tool that was built with the major types of skin cells, which are shown in the clusters in this UMAP, where the UMAP allows this reduced dimensional of the gene expression data to 2D that is easier for visualization. And this tool also allowed us to map our engineered cells to this UMAP to determine whether any of them resembled SIBO in gene expression, which a few did, as you can see that the SIBO is colored here. Our approach also allows us to determine which TF drove the differentiation, so the combination of TFs can be refined. However, it is important to note that one limitation with this approach was that the timing and the level of TF induction could not be controlled. To conclude this section, the direct activation of TFs resulted in cells that are low in stemness, indicating differentiated cells. And a few of these engineered cells resembled human SIBO in terms of gene expression. And once this model is finalized, this could be significant for future sex variability studies, where we can have a human model to study the cellular and molecular genetic aspects of sex and their interactions with neuroscience. In the future, it could also be applied by obtaining cells from sex-gender diverse individuals to improve their gender-affirming care. Moving on to dissertation MB's background. So, after looking at the cellular level of sex, another level of sex is behavior. And there has been a gargantuan amount of work on neuroscience of sexual behavior. A lot of it were conducted on rodents. In general, scientists have grouped these three phases of rodent sexual behavior, beginning from the appetitive, where the rodents investigate each other, detect, and approach. Then there is the action phase, which is also known as the consumatory phase, where one of the rodent would exhibit lordosis, which is this arcing of the back that allows the partner rodent to mount or intramit on top in this position. And this is followed by ejaculation, where the rodent would remain stationary, and often you would see them falling from the back like this, which indicates ejaculatory behavior. And that's when they enter into this refractory phase. So while scientists have observed in most studies that rodent sexual behavior depended on gonadal steroid hormones, in some circumstances, many species exhibit sexual behavior independent of gonadal steroid hormones. For example, since the 1960s, a type of mice called the B62F1 hybrid mice are known that after castration, a proportion of them exhibited ejaculatory behavior over 1 year after castration. Neuroscience studies have since been performed on these B62F1 hybrid mice, and these types of studies entail behavior analysis, which has been done by manually annotating video recordings of these behavior, which could typically accrue to over 1,000 hours in a standard experiment. So you can imagine this type of analysis being very time-consuming. That's not all, but the rigorous annotation of all the sexual behavior parameters that are currently used in the field would take 10 to 20 times longer than the video duration, making it extremely labor-intensive. In addition to that, the different individual annotators are also known to differ drastically to each other. So these are some of the reasons why people have started to pivot towards a more automated way of doing this. And one popular way is post-estimation, where you can get the machine to predict the locations of the body of selected body parts like these. Then the kinematic information can then be used for the machine to learn the behavior. In the case of supervised learning, you can directly tell the machine when the behavior is occurring so that it can learn it. So in this section, I aim to automate the analysis of steroid-independent rodent sexual behavior that paves the way to construct the neuroscience around this phenomenon. So to do post-estimation on our videos, I train the machine to recognize body parts using the post-estimation tool called Deep Lab Cut. And this is an example video of the machine predictions in a video that the machine has not seen before. And this graph, what it's showing is the performance of this model in terms of the machine errors in pixels when compared to the ground truth, which is where the body parts are meant to be based on our labels. And these errors are relatively low considering the size of our videos. So for the next step is for me to be able to get the machine to recognize the sexual behaviors, which a few of them are shown in this video, where you see this intromitting behavior. Then you see it kind of stay stationary and then force off the back, which is the ejaculatory behavior. So in order for me to get the machine to recognize the behavior, I could feed the machine these body part relationship information and then tell the machine exactly when the behavior is occurring so that I could train classifiers to predict these behaviors. So how the classifier prediction works is that it would generate the probability in each video frame to which how likely does it think that the sexual behavior is occurring. Then from that probability, I could set a threshold to get the machine to output data that says that the behavior is occurring if it is above this probability threshold. However, these probability data has a lot of noise, so I also smooth the data by considering the set amount of consistency the machine detected that behavior in a selected number of subsequent frames. And this is one example from one of the combination of the parameters I mentioned. It is a low resolution video, so you can't actually see the text encoding, but just keep in mind that when it's flashing red, it's predicting mounting or intromission, and when it's flashing orange, it's predicting ejaculation. So starting, they are separated, so the machine hasn't predicted anything. And you see now it's mounting, intromitting. Now they're separated. Still separated. And then you see it mount, intromit. And then you kind of see it stay stationary. So this is where it exhibits ejaculatory behavior. And it would stay in this position for a little bit of time. Then it separates and the machine stops predicting. So for me to quantify exactly how well the machine did, and this is an example result for the ejaculatory behavior, this is the result only for the ejaculatory behavior. To quantify how the performance went for this classifier, we manually annotated 10 videos that the machine has not seen before in terms of the behaviors of the ejaculatory behavior, and this is treated as the ground truth. Then I compared the machine predictions with these different parameter setting combinations. Then for each combination, I measure the recall, which accounts for the false negative, and the precision, which accounts for the false positive, and the F1, which is a combination of both. All of these metrics are out of 1. So you see, while some of them seems to be higher, it was very difficult to balance the precision and recall. And this indicates a lot more future work is needed in order to get it up to a standard that we would like to see. Another limitation to this approach is that it relies on post-estimation. But as you may imagine, some of the behaviors may not be able to be captured sufficiently in this way. To conclude this section, the machine could predict body part locations of the mice dyads, and the machine could predict a few sexual behaviors, even though the classifier needs to be improved. In terms of the significance, once the model is ready for future sex variability studies, this could facilitate experiments to construct the neuroscience relating to steroid-independent rodent sexual behavior. And in the future, this type of research could be applied to develop non-hormonal strategies that addresses sexual behavior health in sex-gender diverse communities. So next, I am going into dissertation MC's background. So, in the previous section, I studied sexual behavior as single dyads. But across the tree of life, there are diverse types of sociality, including different mating systems, that indicate sexual behavior occurs in a collective context in many species. An example is lacking, which occurs in several species, including the black bucks shown here. And in lacking, this is where you have a proportion of the species defending a territory like this. Then there are many of these territories, and they are aggregated in these close clusters. And on these territories, you can see the mating displays are performed to entice visitors and aggression to chase away other visitors. And people have postulated this as a collective behavior because these individual interactions, they give rise to this emergent pattern in the collective, such as through their movements, through the mating interactions that would then form and maintain these legs. In collective behavior, constructing the neuroscience cannot rely on studying isolated brains, as shown in this study, where they were looking into the frontal cortex of the bats, and they found that the neurons here were sensitive to the identity of individual bats that allows the interaction to be tailored to specific partners. And the neuron sensitivity was also more precise towards the partners that were more close by compared to those that were more distant. During these social interactions, they found that the frontal cortex becomes synchronized, and this was extended across all of the bats that are listening to the call, promoting this collective brain coupling. So what this may suggest is that studying lacking may benefit from studying multiple brains simultaneously to gauge the interactions at the neural level. One suitable species that could be of interest to study the collective brain relating to sex variability are these Astatotilapia burtoni, which are East African cichlids. And this is because they also exhibit lacking. Not only that, but in the past decades, a growing number of neuroscience studies have been conducted on them. You see in this diagram that these cichlids also exhibit this social hierarchy of dominant and subordinate cichlids, where the dominant are very colorful compared to the subordinates, and that they defend this territory in the lac. What's also interesting is that they exhibit this mouth brooding, where eggs are fertilized in the mouth and kept in the mouth until the young fish are released. It is also important to note that this dominant-subordinate hierarchy is not a binary but a spectrum. It is dynamic where social ascent and descent frequently occurs. So in this section, to facilitate studies on multiple brains to study sexual behavior in a collective context, I aim to automate behavior analysis in cichlid communities. So, Professor Peter Dijkstra's lab in Central Michigan University has done many studies on these East African cichlids, and he very generously provided the video data of these cichlids in different setups, including some that had up to 24 cichlid in a tank. So in these videos, Each cichlid were labeled with selected body parts and then trained for pose estimation, again using deep lab cuts. And this is an example of how the machine predictions performed to predict the body parts in this 24 cichlid tank. And this graph shows the performance of the pose estimation model, which is quantified in errors in pixels that the machine had when using the manual labels as a ground truth. This error for the test data was particularly high for the pictorial fins, considering the size of our videos. And this was expected because these fins, these pectoral fins, which are on the side of the fish, cannot always be easily seen in these videos, and therefore it cannot be consistently labeled. Individual identification is crucial to ascribe behaviors to a particular individual and to determine which cichlid is interacting with which other cichlids. Unfortunately, the individual identification was not yet achievable in our current model. And you're going to see this in this example. Just to keep in mind that the color of the poses indicates each individual that the machine is predicting. So in this video, you're going to see the pose colors to be changing frequently, indicating a failure in individual identification. So while I could not do individual identification by the machine, I could still try it on this setup, where there are two cichlids and they are separated in the tank by the divider, because I could define the poses of an individual based on their location in the tank. So I manually assembled the poses to each individual based on their tank location, like this. Once I could identify specific individuals with the pose data, I could then obtain the kinematic information of these poses. And this is a result for one of the videos that I did, where I calculated the speed of the pose based on the pixels that were moved per second for each video frame. And to prevent cluttering of this graph, I am only showing these three body parts. So in this graph, you see there are some slight discrepancies between body parts, and that's because not all of the body parts would be visible in different video frames. Then for each body part, these were averaged across the entire video to get the average speed. To conclude this section, I automated the tracking of multiple cichlids in a community. In a setup where two cichlids are separated, I also analyzed the kinematic of individual cichlids. Once the individual identification is successful and the machine learns the behaviors of interest, it could be significant for sex variability studies, where multiple brains could be investigated in sexual behavior in a collective context, such as lacking. In the future, this could also be helpful for improving animal ethics by incorporating sexual behavior as part of the animal welfare. Finally, I go now into dissertation MD, starting with a background. So one of the limitations with these cichlid studies was that these were conducted in the lab. And what I'm showing in this diagram is how animals in the lab and in the field could differ drastically. For instance, in terms of the interactions with other species, environmental variables, and behavior affordances, which is this opportunity to exhibit diverse behavior. So captivity is known to affect both sexual behavior and the nervous system. And while substantial values has been attained from model organisms in the lab, we equally need to consider the values of minimally captive, free-behaving wildlife could provide neuroscience, including in terms of sex variability and diversity across the tree of life. And one way wildlife has been studied is through camera traps. And these are cameras that can be triggered automatically without human presence. And it has been widely used to study biodiversity, animal activity, and abundance that could be helpful for neuroscientists to ascertain if a species is suitable for studying. I'm showing here are just some example camera trap images relating to sex diversity. Camera trap data usually accrues millions of images, so AI has been used to automatically screen for images with species of interest to different researchers. But the screening based on the presence of behaviors of interest has been very challenging. So this is where post-estimation could again come in, because we could get the pose relationships that relate to sex diversity and use that to try to screen for behaviors of interest. So in this section, I aim to develop strategies to screen sex diversity in camera trap data that could inform which wildlife would be suitable for neuroscience studies on sex diversity. So far, I've talked about post-estimation for specific species and setups. However, there has been advances that models could be generalized to a wider range of environment and species. And this is exactly what deep lab cut offers, such as this one, where it could work across a large range of quadruped animals. So this model could directly post-estimate on a range of quadrupeds that are shown here in the side view, as well as some of their facial features, without providing any more label data. However, since this model was not trained on camera trap images, it performs really badly on camera trap images. Therefore, to improve this model, I curated over 400 camera trap image crops of diverse range of quadrupeds from 3 datasets. 2 of these are from the camera trap repository, and this one was from the camera trap course I participated in Kruger National Park in South Africa last year. So I also included this as part of my dataset. Then the curated images were then labeled with the quadruped body parts, as you can see in these crops as these crosses, which are the body part labels. And the good news is that Deep Lab Cut just released last month their version 3.0, now with features that allows people to fine-tune these super animal models. So future work could use our label data to improve the model for camera trap purposes. As camera traps are used for screening behaviors of interest and to learn more about a species, there are many other considerations that must be factored in for designing and conducting neuroimaging studies on minimally captive, free-behaving wildlife animals. Therefore, I wanted to develop a framework as a resource for neuroscientists to use for carrying out these type of studies. And with that in mind, we brainstormed on the basis of these 10 different questions, spanning from the ethics, the politics, indigenous communities, sex, the practicality aspects of the study, infrastructures, etc. And this is what we came up with. And it is difficult to read and there's no time to go through it, but it is available on my GitHub for those that are interested. But essentially, these were brainstormed from reading through the relevant literature, and 10 questions were constructed for each of these 10 branches, though many are interconnected and overlaps between branches. It is also not exhaustive, but instead is meant to be dynamic, to be updated without asserting an universal, singular static solution. Rather, it is meant as a catalyst to foster discussions and reflections with the relevant communities, not just scientists. Relevant to that, it is important to note that this framework was not drafted in consultation with these communities, including the indigenous peoples, scholars from other disciplines, and the broader LGBTQ2S+ community. To conclude this section, I curated camera trap dataset with quadrupeds that were labeled for body parts that could be used to fine-tune the quadruped post-estimation model in the future. I also developed a framework to assess the feasibility and ethics of neuroscience studies on minimally captive, free-behaving wildlife animals. And this is significant for sex diversity studies to examine the neuroscience in sexual behavior across the tree of life. In the future, it could help inform conservation efforts given the importance of sex diversity to the ecosystem. So coming to an end of my presentation, I will now briefly summarize each aim. First, to study the cellular aspects of human sex, I engineered cells that resembled gene expression of human sebocytes. Then I moved to the behavior aspects of sex and developed classifiers for steroid-independent rodent sexual behavior. Following that, I explored the collective aspects of sexual behavior by developing machine predictions of body parts in cichlid communities. Finally, moving beyond the lab into the wild, I created a dataset that could, in the future, be used to filter for behaviors of interests in camera trap images. I also developed a framework to help design neuroimaging studies in the wild. And these expanded across the scale of sex-related variables, as well as the diverse participants of the more than human world. So my dissertation was an exercise to imagine a world that is different to the status quo by appreciating sex gender as non-binary and non-essentialist, by acknowledging science as a diffractive process that is only one of many partial and valuable ways to construct sex gender, and emphasize the application of the research to benefit sex-gender communities. Our current society is heavily revolved around sex-gender. Therefore, it is paramount that the research on sex variability and diversity aligns with a more equitable and just world.